biologist. Today I'm going to talk about the history of the cell theory. So settle in for story time with Mrs. Dibdin. Until the end of the 16th century, microscopes did not exist. Cells had never been discovered, and one of the accepted theories was the theory of spontaneous generation. This theory predicted that living things could arise from inanimate, non-living material. This theory was old, as in the time of Aristotle, and was based on the observation, for example, maggots appearing in fly larvae appeared on rotting meat that was exposed for a period of time. In the 1500s, this theory was being challenged, but not until the 1600s scientists suggest that the flies that visited the meat contributed to the appearance of the maggots. Reddy, in 1668, performed an experiment that tested this hypothesis successfully, showing that maggots only appeared on meat that had been exposed to flies in the environment. If meat was covered, no maggots were generated. This was one of the first recorded examples of the scientific method to disprove a theory. Like many great discoveries, there was a change in understanding. In microbiology and the discovery of cells, this was the use of the microscope. Before the compound microscope, scientists used magnifying glasses to look at small objects. The compound microscope design uses two convex lenses, one over the other, making the magnification compounded or multiplied. This means that the first lens magnifies the object and the second magnifies that image. Two Dutch lens makers, a father and son, the Janssens, are credited for making the first microscope in 1590. They had a very simple design where they included a wooden tube, and at the bottom of the wooden tube was one lens, and at the top, at the optimized length, was the second. It was super simple and could be held in one hand. These tubes could magnify objects 3 to 9 times and could be held in hand and form the basis of our first compound microscopes. However, they didn't call them microscopes then. From here, the microscope technology continued to improve and increasing magnification from 930 to 150. This improvement can be attributed to the increase in lens quality and understanding of lens shape. A good quality lens has high magnification as well as high resolution. We're going to explore those ideas more as we look into the microscope later. The convex shape of a lens allows it to magnify an object, but the shape has to be grinded out of glass. Both the quality of the glass as well as the way in which the glass is ground to minimize imperfections play a role in determining the lens's clarity. Hooke in England, Leeuwenhoek in Holland, and Galilei in Italy all made contributions into improving the design of microscopes. Hooke's compound microscope was progressive for its time because it used a fine adjustment knob to move the tube holding the lens up and down. His microscope also had a light source to illuminate the specimen. Both of these things helped in understanding the clarity of images. In 1665, he produced a book where he described his observations of living tissue using a microscope. It was in this book they used the term cell to describe the honeycomb elements that he saw in cork. He was looking at dead plant material that had no clear contents and resembled the small compartments similar to the cells of monks. His findings were well respected but not universally accepted by scientists at the time. This is because of the low quality of the lenses, distorted images, giving them a rainbow fringe around the outside, and many scientists were skeptical about the artificial images that were created. Leeuwenhoek was known for his high quality lenses, and this is based on his grinding technique, which was far superior to his contemporaries. He was able to produce lenses that had a much higher resolution. As a result of this work, in 1683, he created crystal, quartz, and even diamond lenses and developed a simple microscope that actually uses one powerful lens that could magnify up to 300 times, perhaps even more. That lens didn't have the typical issues that others did, but unfortunately, he did not record his technique, and so similar lenses could not be produced after his death. However, he was credited with discovering bacteria from his descriptions, and he may have even seen nuclei. Robert Brown is a Scottish botanist and is known for his discovery of cell nucleus. Although his first described nuclei seen on the outer layers of cells of an orchid plant tissue, he discovered that the nuclei are present in a wide variety of plant tissues that he studied. He had no idea at the time the importance of the nucleus or its function in cells. Microscopes are an instrument now that are considered highly valid in microbiology. But after 200 years, it took for scientists to accept that they were useful. It required there to be an increase in lens quality and other technical aspects that allowed for the images to be less distorted and more accurate. The introduction of powerful light sources and precise focusing screws, increasing the precision of the instruments in general, improved the validity of the technology. It decreased the suspicion of the artificial images and observations, and were generally accepted as valid scientific evidence. Shortly after this time, the cell theory was proposed. Schwann and Schleiden in 1838 were scientists discussing the results of their microscopic studies of living things. Schleiden, a botanist, described the regular placement of nuclei that he observed in plant cells. Schwann, a zoologist, recognised the similarity in animal cells that he'd been studying, and they both went to Schwann's laboratory to look at his slides. It was the first time that a common basic structure of all living things had become evident. A year later, Schwann published a book on plant and animal cells, listing three main conclusions, two of which are still rarely accepted today as the basis of cell theory. Schwann's two first conclusions are summarised as: the cell is the unit of structure of all living things. Number two, the cell as a distinct entity as the building block in the construction of organisms. Virchow's studies led to the statement, where a cell arises, a cell must have previously existed. From this, the third statement of the cell theory is derived, that all cells come from pre-existing cells. Virchow had not only discovered cell division, but by implying living things could not arise from non-living elements, he had convincingly also refuted spontaneous generation in 1879. Walther Fermi confirmed Virchow's observations, named the process of mitosis, which we're going to look at in more detail later. The statement that all living things are made of cells forms the basis of what is currently termed the cell theory. The historical development of the cell theory is clearly linked with the story of the invention and development of the microscope. Improvements in the design and use of microscopes, as well as the progress of microscopic techniques to prepare specimens for viewing, play a significant role in advancing our knowledge and understanding about the cell theory. To study the historical development of a theory, we need to know the current view as well as what the view was in the past. New ideas are often linked to advances in technology, which allow for new discoveries to be made. The cell theory forms the basis of all biology, and it states three things. All things are made of cells. Cells are the basic and functional units of organisms, and all cells come from pre-existing cells. However, as we know from the story before, it has not always been the clear biological view. A scientific theory is a broad general idea of explanation provided by scientists and related to observations, and is supported by a large amount of evidence. But it isn't fact, and it cannot necessarily be proved. It can only be supported or not supported by evidence.
Theories are tested by examining whether the consequences, the predictions, are supported by observation and experiment. They build up to the proposal of cell theory in an interesting weaving story, and hearing this historical account, we as scientists should look for evidence that has been gathered to validate the theory before we generally accept it. Thank you for listening again, and hope to see you next time.